Hey students, it's Mr. Lucido. Just wanted to record this lesson again from this morning in case you missed anything or needed to go back and rewatch it. So, in this lesson number 3, there were 2 objectives. The first was just to give a quick review of cellular respiration that you've seen multiple times, but sometimes we need to see it a couple times to help it sink in. So I'm going to go over that real quick. And then the second thing, I want to spend most of this time talking about the 2 phases of photosynthesis. So let's start with cell respiration, just like we did this morning. So what do we know about cell respiration? Well, we know that it occurs in the mitochondria, which is a membrane-bound organelle found in eukaryotic cells, both animal and plant cells is where we'll find it. And it's got this structure that we see in this diagram where it's got an inner and an outer membrane. We know that cell respiration is an exergonic process, which we can use a free energy diagram to represent that, where we have the reactants being glucose and oxygen having a higher free energy than the products, which are carbon dioxide and water. And so that difference in free energy between the reactants and the products results in free energy being released into the surroundings, which through a process that we'll see in cell respiration is used or coupled rather with ADP phosphorylation to make ATP, which stores energy for later use in the cell. Okay, now what we learned about in cell respiration was that there are 3 major phases with a 4th kind of in-between phase. We've got glycolysis, which occurs in the cytoplasm of a cell, and we have the link reaction, which is kind of that in-between phase that connects glycolysis with the Krebs cycle, and it also occurs as the products of glycolysis are entering the mitochondria. That result of the link reaction is acetyl-CoA, which enters the Krebs cycle, which occurs in the inner matrix of the mitochondria. This is our 3rd phase. And then we've got at the very end on the membrane, the inner membrane, we have a process called oxidative phosphorylation, which has 2 parts. We've got the electron transport chain and ATP synthesis by chemiosmosis that we'll talk about in a moment. So those are the major components of cell respiration. Now in your notes, it asks you, does oxygen get oxidized or reduced in cell respiration? So what we're going to see is that oxygen gets reduced throughout cell respiration. In particular, it gets reduced at the end of cell respiration, and we'll loop back to that on the next portion here. Okay, so then let's look at the phase end. So we've seen these models before. You can find the video that I made right before break where I went through this a little bit more in depth, summarizing how these models are connected. You can find that on Schoology and in our study guide. But just so that we have this fresh in our mind right now, what I talked about in class was that we have glycolysis represented here in model 1, the result of which we get pyruvate. Now, throughout the conversion of glucose into pyruvate, we have to use a little bit of ATP, but in the process, we gain some NADH, which are these high energy electron carriers, and some ATP as well. And so overall, we have a net gain of 2 ATP and 2 NADHs. Now, after pyruvate goes through the link reaction, it'll enter the Krebs cycle in the form of acetyl-CoA. And this is the main phase of cell respiration where we rearrange that pyruvate or acetyl-CoA in this cycle through a series of exergonic reactions, which are coupled with either the reduction of NAD+ to capture that high energy electron, or the reduction of FAD into FADH2, another high energy electron carrier, as well as we do get just a little bit of ADP phosphorylation as well. Okay, now all of these steps where we're collecting high energy electrons in the form of NADH or FADH2 are going to make their way to the electron transport chain. And again, this is part one of oxidative phosphorylation, where we've got those high energy electrons being donated to this series of proteins that are embedded into the membrane, the inner membrane of the mitochondria. And as those high energy electrons move through those series of proteins, they're actually going to provide the energy that's needed to power the active transport of hydrogen ions against the concentration gradient, which in this case goes from the mitochondrial matrix into the inner membrane space. So active transport is powered by the energy of the high energy electrons passed along in this transport chain. Now, what this does is it creates this constant gradient, this constant chemical gradient, where we've got a high concentration of hydrogen ions in the intermembrane space and a low concentration in the matrix of the mitochondria. And that allows for the second part of oxidative phosphorylation, which is chemiosmosis, to occur. And chemiosmosis, the main protein involved here is ATP synthase, which facilitates the diffusion of hydrogen ions through the membrane, down the concentration gradient. But in so doing that, it actually provides the energy that's needed to phosphorylate ADP. And this is where we're going to get lots and lots of ATP made. And so if you're looking for how is chemiosmosis related to ATP synthesis, chemiosmosis refers to the movement of, in this case, hydrogen ions down the concentration gradient for the purposes of powering ADP phosphorylation. Okay, and just to wrap things up here, I mentioned at the very beginning that oxygen is reduced, meaning that it gains an electron. And where does that happen? Well, it happens right here at the end of the electron transport chain. Those high energy electrons that are now spent are gained by oxygen and combined with a few hydrogen ions to make some water. So oxygen is reduced during cell respiration. Okay, so that is a quick review of cell respiration. Again, my biggest recommendation is study these models from your cell respiration group work, and you'll have a good understanding of the details that you need to know for cell respiration. So now let's photosynthesis. So photosynthesis is this other big process that happens in biology, and you're going to see some similarities with the concepts we learned in cell respiration, but also some unique concepts as well. A couple things that we need to know about photosynthesis. One is that it occurs in the chloroplast, another membrane-bound organelle in eukaryotic cells, specifically plant cells, that, similar to mitochondria, have a dual membrane, an inner and an outer membrane. However, the chloroplast also has some slightly different structures. It's got these thylakoid stacks. There are these little membrane-surrounded stacks that are arranged on top of one another. You have, in between the thylakoid stacks, the space is filled with a substance that we refer to as the stroma. That's going to be relevant for the location of one of the reactions we're going to talk about. But chloroplasts are the site of photosynthesis. We see that photosynthesis overall is what we refer to as an endergonic reaction, because we are taking in large amounts of energy in order to rearrange reactants that have a low free energy into products that have a high free energy. In this case, most of that being stored in glucose. Now, in this case, water, it asks, does it get reduced or oxidized throughout photosynthesis? Well, we see from this diagram that it becomes oxidized, meaning that it loses electrons, and we'll see where that happens in a moment. But water gets oxidized throughout photosynthesis. Okay, now there are two main phases that we're going to talk about in photosynthesis. that are summarized right here. We've got the first we call the light reactions or the light dependent reactions that occur in the thylakoid stacks of the chloroplasts. And then we also have the Calvin cycle, which occurs in the stroma, which again is just the space in between the thylakoids. Okay, so let's talk about each of those two phases a little bit more in depth to get a better idea of what happens in synthesis. So let's start with the light dependent reactions. Okay, so the light dependent reactions, that first phase that is occurring inside of the thylakoid stacks. The main purpose of this light dependent reaction is to harvest energy from the sun in order to excite electrons that can be used for the creation of ATP as well as NADPH, which we'll talk about in a moment. Okay, now a couple of things we got to know. There are some key molecules involved in the light dependent reactions. What we have are two photosystems. We got photosystem 1 and photosystem 2. And we also have proteins arranged in an electron transport chain similar to what we see in cell respiration. Again, this is all happening on the membranes that are surrounding the thylakoid stacks. Now, if you take a little bit of a closer look at a photosystem, we've got a couple of diagrams down here. It's actually pretty impressive what we see in a photosystem. So here is photosystem, I believe 2. This photosystem, you can see, has lots and lots of subunits. It's a large protein complex that's got a bunch of, you can see, alpha helices, beta plated sheets, and embedded in this protein are actually these molecules called pigment molecules. And pigment molecules, which are indicated in green here and here, there are multiple types of pigment molecules inside of a plant. One of which you've likely heard of before is called chlorophyll, and there are kind of two forms of chlorophyll. We got chlorophyll A and chlorophyll B, but then we also have a series of pigments called carotenoids, which are slightly different but perform the same function in that they help absorb solar energy. So taking a closer look at the structure of a pigment molecule, this is actually chlorophyll A right here. And if you notice, it's got a familiar structure. It's got a hydrocarbon tail, which means that it's got a hydrophobic region right here. And this helps it to interact with the hydrophobic regions of the thylakoid membrane, as well as the hydrophobic R groups of the photosystem protein. But what we also see is that the head of this molecule has a light absorbing region. There are specific properties of this region that has a magnesium at the center that allow it to absorb the energy from a photon that comes from solar energy, and pass that energy along throughout the photosystem in order to ultimately excite electrons into the electron transport chain. So before we talk about how that's done, if you look at this diagram here, the differences between those different pigment molecules is actually what type of light they're best at absorbing. Chlorophyll A tends to absorb light closer to this end of the visible light spectrum, so about 425 nanometers. Chlorophyll B is closer to about 475, and then carotenoids about 500 nanometers. Now, this visible light spectrum, if you've seen it before, we've got different colors throughout this visible light spectrum that each contain different levels of energy stored within them. So we've got a variety of pigment molecules, often in a plant, in their photosystems, because it allows them to absorb a broader range of visible light or energy in visible light. And so it's advantageous for a plant to have multiple types in order to be able to absorb sufficient energy throughout different seasons, different environments, different conditions. So that's what we'll see, multiple types of chlorophyll or carotenoids in the photosystems. Okay. So looking a little bit closer, this diagram right here gives us a little bit of an outlined form of the photosystem. So again, we've got chlorophyll that's embedded in this protein complex that absorb photons and then are able to transfer that energy between other chlorophyll or pigment molecules to a central region where that energy is used to excite electrons as they move into the electron transport chain. Okay, now where are those electrons specifically coming from? Well, so this is where water gets oxidized. Remember, we mentioned at the beginning that water gets oxidized because it gets split into oxygen and hydrogen ions, and as a result loses multiple electrons into this photosystem, which are then excited through the absorbed energy by the pigment molecules to a high energy state and then enter the electron transport chain. So unlike cell respiration, where the electrons are already excited when they get to the electron transport chain, in this case, we have to excite the electrons using absorbed solar energy before they enter the electron transport chain. Now, this is the part that will likely be familiar. As those electrons move through the electron transport chain, that energy, again, is used to pump hydrogen ions across their concentration gradient, in this case, from the stroma into the thylakoid, like we see right here. Okay, now as that occurs, energy is lost from that high energy electron, and what happens at the end of this electron transport chain is that photosystem 1 has a very similar structure, is also absorbing photons in the chlorophyll pigment molecules and other pigment molecules, will then re-excite those electrons to a high energy state. And instead of entering another electron transport chain, those will actually be captured by a high energy electron carrier that's similar to NADH, but in plants it's NADPH. Now that high energy electron carrier is going to head to the Calvin cycle, where it's going to be used for different steps in the Calvin cycle. Now, what about the hydrogen ions that are being pumped? Well, just like in cell respiration, that concentration gradient is used to make ATP. There's an ATP synthase protein embedded in the thylakoid membrane as well, where those hydrogen ions can be facilitated out. But as that occurs, provides the energy to phosphorylate ADP into phosphate. So very similar to what we see in cell respiration. Okay, now at the end of the light-dependent reactions, right, we've used up some water, but we have NADPH and ATP as products that we're going to use to power the Calvin cycle. Okay, so that is the light-dependent reaction, and we're going to see how it's connected in the Calvin cycle in just a moment. Okay, so the second phase is called the Calvin cycle, or the light-independent reactions. Now, what we're going to see for the Calvin cycle, which is occurring in the stroma, is that these are a series of endergonic reactions that are going to be powered by the energy-carrying molecules that were produced in the light-dependent reactions. Now, there are three phases of the Calvin cycle: carbon fixation, reduction, and regeneration of something called RUBP. Okay. Now, at the very beginning of the Calvin cycle, we have our carbon dioxide, which is a reactant of photosynthesis that's entering the cycle, and it is fixed to a molecule called RUBP. It's fixed onto this molecule. Now, what this creates are these very unstable molecules that have 6 carbons. And you can see, right, RUBP has 5 carbons, and we're adding 3 carbon dioxide, so a total of 3 carbons, giving us 3 of these 6 carbon molecules. Now, this molecule is very unstable. It's very difficult to fix that carbon onto the RUBP. And so because of that, we have an enzyme called Rubisco. And Rubisco is this pretty amazing enzyme that is actually thought to be the most abundant protein on Earth that facilitates this or catalyzes this reaction in order to allow carbon fixation to occur. Now, throughout the Calvin cycle, what we see is that immediately that 6 carbon molecule breaks down into 3 carbon molecules called phosphoglycerate that then have to be reduced through a series of endergonic reactions into a molecule called G3P. Now, in order for that to happen, you need to add lots of energy. And where's the energy coming from? Well, it's coming from the ATP and NADPH that came from the light-dependent reactions. And as we see, what happens is we get a total from 3 carbon dioxide molecules, we get 6 G3P molecules. One of those actually exits the Calvin cycle and will be combined with other G3P molecules or other molecules in the cell to make organic compounds such as glucose or other sugars. But the rest of the G3P molecules are recycled, and they're used to actually regenerate RUBP molecules. And if you actually count the carbons, we have 5 G3Ps remaining, which makes 15 carbons. And RUBP has 5 carbons itself. And so we're able to regenerate 3 RUBPs with the 15 carbons that we have. And that's a very endergonic process that requires ATP in order to allow that to happen. But that allows the Calvin cycle to keep on going. So what we see in the Calvin cycle, again, are 3 different phases. We see the production of G3P, which can be used to create glucose, but we also see the regeneration that allows the cycle to continue. Again, the connection between the Calvin cycle and the light-dependent reactions is that the light-dependent reactions provide the energy molecules that are used to fuel this Calvin cycle, which contains a lot of endergonic reactions. Okay, so I know that was a lot. If you want to look more into these 2 processes, you can check out sections 10.1 to 10.3 in your textbook, although the details that I've went over in this lesson are the main ones that you need to be familiar with. And again, we'll get some more practice with the group work during next class. So reach out if you have any questions, but I hope that clarified it for you, and I will see you soon.